Hello student. Today we start the BSc second year paper 7 Fish Developmental Biology. BSc second year paper 7, that is Fish Developmental Biology. Within that already we are completed unit number 1, unit number 2, unit number 3, and the last one, that is the unit number 4. Within that unit number 5th, last one, that is methods for age and growth determination in fishes. The topic is that method of age and growth determination in fishes. Before that, the age and growth determination, some following aspects I will give. To find out the year classes which supports the fisheries, then to estimate the mortality rate, then to find out the age of a particular species at sexual maturity. Okay, this is all the basic concept of method of age growth determination in fishes. Then to ascertain the minimum size and age of species for fishing, then to compare rates of growth in different bodies of water to identify favorable and unfavorable condition, then to estimate yearly fluctuation in the fish population of a certain body of water. All are the aspects to explain the method of age and growth determination. Right. Before that, the some concepts with the help of that I will explain about the method of age and growth determination in fishes. Number one, that is the tagging method. What is meant by the tagging method? Fish are marked or tagged. It means that tagged is nothing but the marked one. Fish are marked or tagged and released after recording initial measurement of length and weight. But I'm saying that recording initial measurement of length and weight, the fish are recaptured after an interval of a few months and measure again. Okay, the growth rate is calculated from the change in size over the time period, and means fish spent in its natural habitat. Marking should be done in such a way that it does not affect the behavior or feeding habit of fish. When you are tagging that time, it does not affect the behavior or feeding rate of the fish, and it can be done by clipping the fin rays, coloring the epidermis by spray painting, or providing someone's or rings or scales or bows, various type of their the calculation or there for the tagging methods. Okay, tags of various kinds are used and bear a detail about the fish. Data of tagging and are attached to the body of the fish. Date of tagging on which date? Then are attached to the body of fish. A small disc of plastic, aluminium, silver or nickel can be used to tag and attach to the body by means of a thin wire. Means the wire is tight. It is a process. Means tagging method is the process. After encouraging the lower side or caudal peduncle without pairing the body of fish. This is one of the method that is called the tag. Already I told tag is nothing but the marked one. How, what the purpose? For the purpose of growth, for the purpose of age, the method is used. That is called the tagging method. The next one, that is the marking method. In this method, fishes are marked after measuring the length and weight for identification and are released in the natural habitat. Marking and tagging method both are the near about same. After a few months, these fishes are recaptured and measured again. Why? for the purpose of growth, for the purpose of length. The changes in size during the interval gives growth rate. Marking of fish can be done by clipping the fin ray, coloring intestine by the spray, painting, providing means all these things providing by the rings of the scales or the bones. This is a purpose for marking. You can mark in plotting or other purpose. On the basis of that same the marking for the purpose of growth, for the purpose of length, for the purpose of age. Certain is chromium dioxide, fat, fast blue, lead can be injected or served to recognize the fish when recaptured. This is one of the method of that is called the marking method. And next one the scale method. You know the fishes have the scale, dinoid, cricoid, and all these scales. On the basis of that, age and growth rate of fish can be accurately determined by counting the annually of the scale. But what is meant by the annually? We can see scales from a very useful tools for determining the age because rings are formed every year as a fish during growth in age. Ring is formed on the scales. Food is available in summer. The fish feed and grow rapidly. It means that food is available in summer month. That time fish feed and grow rapidly. Then ring formed during this month are widely spaced food supply in limiter during winter. Hence, growth is slow so that the rings formed during the spread close to each other are compact in month of winter. Thus, the scale show light and dark bond on the scale. There is a margin, there is a show that is the light and dark bonds are formed in 1 year that are nothing called that is the annuli. Okay, the number of annuli on the scales indicate the age of the fish. This is also one of the method that is the scale method. On the basis of scale, on the basis of annuli, you can count how many annuli are present on the scale. With the help of that, we can identify, we can search, we can research. That is the age of fish. Then the radiocarbon obtained method. In this method, scales are taken from a live fish and are incubated in a medium contacting the ammonia acid glycine made radioactive by C14. After incubation for about 4 hours, the rate which C14 glycosine is incorporated incorporated into the cell is measured by a level of beta. It is beta radiation limited by the scales. Increases in incorporation of C14 by the scales as shown by uh, silencians counter means faster rate of growth of the fish. This is one of the method that is the radiocarbon taken method. The last one that is the RNA DNA ratio. You know very well about the RNA DNA ratio. This is a method to measure protein synthesis rate, giving an index of growth rate. DNA content is constant per cell, but RNA contained in a function of the cell of protein synthesis. Thus, RNA and DNA ratio show the rate per number of cells in tissue samples. The ratio means the measure the weight of the fishes. However, such compare can be made within the same species. Means what that is the growth in animal is not constant for all year. Okay, the young grow at a fast rate and this goes to decreasing as the animal advances in age until there is no further growth. Okay, the rate of protein synthesis in a cell is measured to serve as an index of growth. Amount of DNA in a cell is constant, but RNA content depends on the cell's protein synthesis. Thus, the ratio of DNA RNA shows the rate of protein synthesis in a tissue sample. The ratio is correlated with the gain in weight in some fishes. What it means that correlated with the gain in the weight in the some fishes. However, such comparisons are possible among the individuals belonging to the same species. I will repeat again. The rate of protein synthesis in a cell is measured to serves as an index of growth rate. An amount of DNA in a cell constant, but RNA content depends on the cell protein synthesis. It is an important point. Thus, the ratio of DNA and RNA show what they show. The rate of protein synthesis in a tissue sample. The ratio is correlated with the gain in weight in some fishes. However, such comparisons are possible among the individual belonging to the same species. Okay.